Welcome back to the research track. I'm very pleased to welcome our last researcher of the day, but come back because we have 3 more tomorrow. But right now we have Dr. Sriram Vaidyanathan, most recently of the Porteus lab at Stanford University. After completing his PhD in 2016, Dr. Vaidyanathan joined the Porteus lab at Stanford to further his interest in applying the CRISPR-Cas9 based methods to develop therapies for monogenic diseases. While there, he spearheaded a multidisciplinary team from 4 separate labs at Stanford to develop an autologous airway stem cell therapy for cystic fibrosis. He is currently working on extending this platform to develop a genome editing platform that restores CFTR function in airway cells affected by any CF mutation, including those that cannot be treated using modulators. His goal is to develop methods to transplant corrected stem cells back into the airway, thus enabling a durable treatment for CF upper airway disease. Today he presents transplantation of gene-edited upper airway basal stem cells in immunocompromised mice using fibrinogen-based scaffolds to treat CF sinus disease. Welcome.
Thank you so much for the really kind introduction. I would like to start by thanking CFRI for this amazing conference and for their support for our research. So today I'll be presenting our work on genome editing upper airway basal stem cells and then trying to transplant them into the sinuses of mouse models to eventually treat CF sinus disease. So I don't think I need to introduce CF to this audience. The only sort of genetic fact I want to remind everybody is that CF is an autosomal recessive disorder. So each patient has 2 mutated copies of CFTR, 1 from each parent. And so when you think about this from a therapeutic perspective, restoring even 1 copy of the correct CFTR gene in a cell should be sufficient to treat CF patients. And as many of you know, CF can be caused by a wide variety of mutations that have been reported in CFTR. So depending on how the mutation affects the production or the folding or the function of the protein, the severity of the disease may be different. So you have mutations that cause highly morbid versions of CF. And then on the reverse side, you also have mutations that cause less severe versions of CF. Delta 508 is a mutation that everybody's familiar with here. It's the most commonly observed mutation and that's observed in about 70 to 90% of CF patients, at least in Europe and North America. And so the remarkable aspect of CF is that over the past 70 years, there's been a 10-fold increase in the lifespan of CF patients. And a lot of this has been due to better ways of improving nutrition in these patients. So first by the introduction of pancreatic enzymes, and then by methods to sort of prevent and treat infection. So the development of better and better antibiotics. So the combination of these approaches improved survival from less than 5 years to about 40 years. And then, but the other point to note here is that these approaches did not directly restore CFTR function. Now that has been achieved using modulator therapies over the past decade, and we're all excited about this. And this has been an incredible achievement, and it's predicted to increase survival in patients dramatically over the course of the next few years. So these CFTR modulator therapies are incredibly effective in preventing lung exacerbations. That said, there is a defined class of patients that cannot benefit from these therapeutics because they have mutations that are not amenable for treatment using modulators. And then there are also patients who show heterogeneous responses. So they have mutations that should respond to CFTR modulators, but don't really respond in a way we would predict. And then there are complications and contraindications associated with the use of modulators. And lastly, they must be taken daily for the life of a patient. So there are some limitations. And so genetic therapies are one modality that hold the promise for a long-term treatment for all CF patients. And as we think about genetic therapies and all CF patients, I just want to spend a couple of slides looking at the possibility of whether we're missing CF patients. So CF, as we all know, has been best characterized in the Caucasian or white population, where about 1 in 2,500 to 3,500 newborns are diagnosed with CF. So, and we estimate about 70 to 100,000 CF patients in Europe and North America. And it's thought to be much rarer in other races. So about 1 in 17,000 African Americans have been reported to have CF, and 1 in about 31,000 Asian Americans have been reported to have CF. And it was at this point we noticed several South Asian CF patients at Stanford, because it's an area with a very high Asian population, and this sort of piqued my interest. And this was right around the time of the pandemic, and so this was a really nice dry lab sort of project, which was done in collaboration with Zach Sellers. So together we requested data from CF registries in the UK and Canada, and we particularly focused on these registries because they classify South Asians. So South Asia is the region around India and Pakistan. So we requested data specifically from registries which classify South Asians separately. And we looked at the prevalence of CF in South Asians versus people from other parts of Asia. And what we found was that about 60 to 90% of Asian CF patients in all of our data sets, so whether we looked at the Stanford data set, the UK data set, or the Canadian data set, about 60 to 90% of Asian patients tend to be of South Asian origin. And so based on this, we estimate a prevalence of about 1 in 10,000 to 1 in 20,000 South Asians having CF. So then the next question that was in our minds was whether these patients were amenable for treatment using modulators. And so we first started by looking at the mutations that affect this population. And we noticed that only 50% of these patients have at least one allele with delta F508. So then the natural extension is whether the other mutations in this population are amenable for treatment using modulators. And so when we looked at this, only 50% of patients in these data sets had 2 mutations that were amenable for treatment using modulators, which means, it's good news for the 50% that can be put on modulators, but it's, it tells us that there's more work to be done in developing therapies that are applicable for the other 50% of this population. Now, this is not unique just to the Asian population. So other researchers have shown that significant fractions of Hispanic and Black CF patients also have mutations that are not responsive to modulators at levels that are higher than what you would see in the white population. And this boils mostly down to the type of mutations that are present in these populations. And I just want to take one further step. So we look at the registry data in Europe and North America, and we always see that there's about 10% of patients that don't respond to these modulators, and a lot of these patients tend to be from these sort of other ethnicities. But I also want to remind everybody that 80% of the world's population lives in Asia, Africa, or South America. So if you really take the prevalence metrics that have been reported in these populations and apply it to the global population, there should be about 200 to 300,000 patients, CF patients in the world. And I would estimate about 40% of these patients would still need therapeutics that are not modulators, or that are new modulators that maybe or new therapeutic modalities that can restore CFTR function in the presence of these mutations that are not responsive to modulators. And so with that, I think there's definitely an interest in genetic therapies which hold the promise for a modulator agnostic approach for treating CF. And the previous talks, there was one from Dr. McRae and Dr. Jane, and then the most recent one from Dr. Jonas. They all talked about different in vivo gene therapy modalities. The one in gene therapy that's been tried in the clinic is what I call transient gene therapies. The idea here is to overexpress CFTR from the terminally differentiated epithelial cells in the airways of CF patients. Now, this has been attempted previously in the clinic, but they did not show durable benefit in these trials. And mostly the challenge that's been recognized is the presence of thick mucus in these patients, which limits the amount of the epithelium that can be successfully transduced. And as you heard from the previous talks, there are multiple attempts at developing new technologies that would transduce this epithelium much more effectively. But that's not the focus of my talk. What we wanted to see was whether there was an alternative solution that we could come up with using genome editing. And so our idea was to take airway stem cells from CF patients, either from the nose and sinuses or from the bronchi, and then gene correct them ex vivo using CRISPR-Cas9 and other reagents, and then transplant these cells back into patients. The advantages here are that we don't have to worry about immune reactions mediated either against Cas9 or any of the viral vectors. You can directly target the stem cells of these organs. And so you could hope for a durable cure. And then you can characterize the product really well before you administer these to patients. The one challenge here is that airway stem cell transplantation has never been done in humans. So recognizing this, we wanted to start with a focus on CF sinus disease. So from a clinical standpoint, CF sinus disease affects most CF patients. And then CF sinuses act as a reservoir for pathogens that have been shown to seed subsequent lung infections, especially in transplanted patients. And from a strategic standpoint, CF sinuses are easy to access. And then ablation of the sinus epithelium is routinely performed endoscopically, and it's safe. And so we thought we could piggyback on this sort of a surgical ablation to then transplant our corrected airway stem cells. And our hope is that we would take the tools developed from this effort to then develop strategies to treat CF lung disease in the future. So our platform looks something like this, where we isolate upper airway basal stem cells from patients, expand them in vitro, edit them using Cas9 and AAV, put them on a biomaterial, and then hopefully transplant them into patients in the future. So when we started this project, we started with a two-pronged strategy. So the first strategy was the Delta F508 strategy, where we showed the correction of the Delta F508 mutation, which affects most CF patients, as we know. And then the second strategy was to insert the entire cDNA of CFTR in exon 1 to restore CFTR function in all CF patients, regardless of the causative mutation. So when we started this project, there had been previous reports where they had attempted to correct CF-causing mutations, particularly Delta F508. So the first ever study using CRISPR-Cas9 to correct a CF-causing mutation was done in intestinal organoids. And then there were subsequent studies done in iPSCs. Now, in both of these studies, the correction efficiencies were low, and then they were using sort of selection methods using puromycin, which are not readily clinically applicable. And then there are challenges associated with the generation of airway cells from iPSCs. That's still a work in progress. It's progressed a lot, but we're kind of still not there yet. And so when we started this project, our question was how do we correct airway stem cells, and how do we achieve 10 to 100-fold higher levels of correction than what had been reported previously? And so before we go too much further, I just want to remind everybody about the different sort of pathways involved in gene correction. I know the previous talks covered it, but just to cover my basis, I wanted to remind everybody. So Cas9 is a bacterial enzyme that complexes to a guide RNA. Now, this guide RNA is what gives Cas9 the specificity. So if a guide RNA matches the sequence specific to the CFTR locus, then that guide is going to enable Cas9 to induce a break in the CFTR locus. Now, this break is repaired using the endogenous repair machinery in the cell. So one mechanism is called non-homologous end joining. I call this the lazy pathway. So the cell assumes that no information is lost here, and it just pastes the break, which results in errors that are insertions or deletions, resulting in a knockout of the gene. So this is not the pathway we want. The pathway we want is homologous recombination, where if you supply a correction template that has homology arms that mimic the site, you can essentially trick the cell into incorporating any exogenous information into that break site. And so for this process, we use Cas9 RNP, so the protein formulation. We use a chemically modified guide RNA, which has been previously shown in the Porteus lab to be more effective at improving Cas9 activity. And then we use an AAV, adeno-associated virus, to shuttle in the homologous recombination template into these cells. So using this approach, we first corrected the Delta F508 mutation in both sinus and bronchial derived stem cells in patients, and that paper was published in 2020. The sort of second project we did on this topic was the insertion of the CFTR cDNA in exon 1. And just to reiterate, we were interested in the strategy because it's mutation agnostic, and it's particularly relevant for patients who are not amenable for modulator therapies right now. The challenge here is that the CFTR cDNA is rather large. And when we started this project, it was unclear if insertion of the cDNA was going to be sufficient to get CFTR expression from the endogenous locus. If you look at the CFTR cDNA, it's about 4,500 base pairs long. And when you look at the packaging capacity of AAV, it's about 4,800 base pairs long. So you don't really have space for including sort of long homology arms that are necessary for efficient homologous recombination. And you also don't have room for a selectable marker. So here we used a split strategy that had been previously developed in the lab, where we used a guide RNA to insert a break in exon 1. And then we have a correction template that codes for the first half of the CFTR cDNA. And then we added in a guide RNA at the end here. And then we have a second correction template that has the second half of the CFTR cDNA. So when the second break happens, that break is repaired using the second half of the CFTR cDNA. And then that template also has a PGK promoter that's driving a truncated CD19. CD19 is a protein that's normally found in B cells. What we've done here is we have truncated the intracellular portion of the protein so that you just have a surface epitope that's going to serve as an enrichment tag. And so the corrected CFTR locus in our strategy would have the corrected CFTR cDNA, and it would also have the truncated CD19 tag that's going to help us enrich for these cells. So as you might imagine, because it's a split strategy, the efficiency of correction is initially low. So we only have about 3 to 5% corrected cells. So here in this example, it's about 3.5%. But then because we have this CD19 tag, we can use flow cytometry or magnetic bead-based separation to then enrich out the corrected cells. And so we tested this strategy in airway stem cells obtained from the sinuses and bronchi of 14 different donors with CF. And in all of these cases, we were able to obtain an enriched population of corrected cells that had at least 50% corrected cells. And so the next question in everybody's mind is whether this is sufficient to restore CFTR function. So to test that, we took these corrected cells and then we differentiated them on an air-liquid interface. And then we used an Osing chamber assay to look at the CFTR function in the airway epithelia generated by these cells. And so just to sort of orient everybody, so here we have an airway epithelium. And then in an Osing chamber assay, we're measuring the transport of chloride ions across this epithelium. So in a normal wild type airway epithelium, you would have a certain level of chloride transport, and this transport is reduced in airway epithelia generated from stem cells from CF donors. And so in the Osing assay, we use certain small molecules that are activators and inhibitors of ion channels to probe for CFTR function. So we first add amiloride, which blocks ENaC channels, and then we add forskolin, which activates CFTR, and then we add CFTR inhibitor, which then inhibits CFTR specifically. And then we look for the drop in the current induced by CFTR inhibitor, and then we quantify and compare this across samples to see if we have restored CFTR function. So in this example here, you have airway epithelia from a CF donor where you don't have a response to forskolin or CFTR inhibitor. And then the cells from the same donor were then corrected and then differentiated. And then when we tested those samples, you can see a really nice, robust restoration of forskolin and CFTR inhibitor function. And so we did this in multiple samples. So here is the summary of all the samples where we tested this in sinus-derived stem cells. And you can see that the CFTR function is very nicely restored, and it's at the level that's comparable to the CFTR function we observed in non-CF controls. And then here you have data from all the bronchi bronchial samples, where again, you have CFTR function that is comparable to the CFTR function that we saw in bronchial cells obtained from non-CF donors. And so all of this work has been previously published. So I just wanted to reiterate that it is possible to insert the complete CFTR cDNA in the endogenous CFTR locus and enrich for these corrected cells. And we do observe a non-CF level restoration of CFTR function using this approach. So then the topic of today is how do we transplant these airway stem cells, at least back into the sinuses, so that we can start with the therapy for CF sinus disease. So when we were thinking about this, we recognized two factors. So one is the need for a delivery vehicle. So in our preliminary studies, when we tried to transplant these cells into immunocompromised mice, we were just trying to squirt the cells into the sinuses, and then the mice wouldn't like the introduction of a foreign material into their nose, so they would sort of try to expel them, and so all your hard work would just be on the table after a few minutes. And so we realized we needed some sort of a delivery vehicle that would hold the cells in place. And then the second question is that of a conditioning regimen. So when you're thinking of transplanting stem cells, you need to get rid of the existing stem cells that are specific to that organ. So if you're thinking of a bone marrow transplantation, you have certain conditioning regimens that kill the existing bone marrow stem cells in that in the patient, so that there's space for the new cells to go and occupy. And so in the case of airway stem cell transplantation, previous studies have shown the use of naphthalene and sulfur dioxide and polydocanol for the transplantation of these cells. However, you can't give a person naphthalene or sulfur dioxide. So we were really also thinking about what are clinically applicable methods to ablate the sinuses so that you can transplant these cells. A lot of the conditioning regimen work was done by Dr. Don Bravo, who's a very close collaborator in the NIAC lab. And then I was focused on screening the biomaterials that we could use to hold these cells in place. So there are, again, different factors you can consider when you're looking at biomaterials. So we considered the ability of these materials to support cell survival and cell growth. And then we also then considered whether they were capable of facilitating tissue regeneration, or at least not hindering tissue regeneration, whether they were compatible with existing delivery technologies, and whether they're clinically compatible, as in whether they're safe, or whether we have to have any special considerations for putting these into a human. And so using these considerations, we identified these materials listed here. And so we evaluated these materials for their ability to support cell survival and growth. And we used Matrigel as a positive control. So Matrigel is a mouse tumor-derived matrix that works really well in vitro, but it's really not clinically compatible because of how it's derived. And so we compared the ability of these other materials to support cell proliferation in a manner that's comparable to Matrigel. And so if you look at this chart here, you can see that fibrinogen and biosilk really support the proliferation of airway stem cells at a level comparable to what you see in Matrigel. And biosilk, just for clarity, is a laminin functionalized spider silk that's commercially available. And then fibrinogen is an interesting option because it's a product that's already used clinically. It's a human clotting factor, so you really don't have any concerns about immune reactions. It's already used in wound sealants, particularly in sinus surgeries. And fibrin collagen gels have previously been reported for making vascular grafts in animal models. And there's a lot of room to engineer gel composition and gelation kinetics if needed, right? And so we wanted to look beyond just proliferation. We wanted to see if the cells maintain their stem-like properties when cultured in these materials. So we looked at the expression of cytokeratin 5 and P63, which are two common markers for airway stem cells. And so you can see both in fibrinogen and biosilk, over 80 to 90% of these cells express both cytokeratin 5 and P63. And the level of the fraction of cells expressing cytokeratin 5 and P63 are comparable to the fraction of cells expressing those markers when cultured using Matrigel. So we were pretty happy with both of these materials. And so then we wanted to evaluate whether these materials work effectively in an in vivo setting. So to test this first, we wanted to manipulate the cells as little as possible. So we found a mouse that has been engineered to express GFP and luciferase in all of its tissues. And we took the upper airway stem cells from these mice. And then Don Bravo looked at different injury models, so mechanical, polydocanol, and sulfur dioxide-based injury, to see if we can transplant these cells using those models. And indeed, when we used Matrigel as our sort of delivery vehicle to test the engraftment of these cells using these models, she found successful engraftment using all of the injury models that were tested here. And so that was really promising. And then because mechanical injury, so we want to use mechanical injury in patients eventually. However, it's really hard to do surgeries in mice because they're really tiny and their sinuses are even tinier. So for the sake of practicality, we used polydocanol for further studies that I'm going to show here. But when we go into our clinical sort of studies in the future, it's going to be a mechanical debridement that's going to be performed by the surgeon. But in this study, I took these GFP luciferase expressing cells from mice, and then I looked at whether fibrinogen-basal could be used to transplant them at a level that was comparable to what we would see in Matrigel. And so you can see here in these pictures that the luminescence that you see in mice transplanted using fibrinogen and basal is very comparable to the luminescence you see from a mouse that's transplanted using Matrigel. And then here the graph is showing the signal over 60 days. So you can see that the mice transplanted using both fibrinogen and Matrigel show stable signal for about 60 days, whereas the mouse transplanted with BioSilk shows a really large drop that really quite doesn't recover. So when we looked at this data, and when you consider the fact that fibrinogen is an endogenous human protein, we thought that is the most promising material to move forward with in our studies. And so but then before we get to that point, we also sacked these mice and so euthanized these mice, and then sectioned the tissue to look at the colonization of the cells, right? And so the first sort of encouraging signal that we got, and so these images were taken by Don Bravo. So the first sort of encouraging signal we got was that we could see a very clear GFP signal in the mouse sinus and nowhere else. So that was really promising. And then when we did the histology, you could see that there's this really nice GFP positive airway epithelium that was generated in the sinus by our transplanted cells. And they do express the other canonical airway markers like cytokeratin 5 and alpha tubulin. So this was really exciting. And so in the next slides I'm going to show you, we wanted to look at the transplantation of human cells using fibrinogen. And so here we took upper airway basal stem cells from human donors, engineered them to express GFP and nanoluciferase, and then we transplanted them into immunocompromised mice using fibrinogen after injuring them using polydocanol. And so here I have data from cells obtained from one donor that were transplanted into 4 different mice. So you can see in all of these 4 different mice that there's a really nice strong signal on day 6. And this signal continues to persist for about 55 days in all of these mice. And so the other mice were sacked at that point, but then the one mouse that did survive that was kept alive till 120 days also showed a strong luciferase signal at the end of that. So that was really promising. And then we repeated these studies using cells from 3 other donors. And so here you can see in all of these donors, these cells show robust engraftment for anywhere between 60 to 130 days. So this is really promising. And so we're in the process of collecting the tissue from all of these mice and looking at the organization of the transplanted human stem cells in the sinus cavities of these mice. So to summarize, what I've shown you today is that we can use genome editing to replace the entire CFTR cDNA in airway stem cells, and that this does result in a non-CF level restoration of CFTR function in vitro. And then we have strong proof of engraftment in our preliminary studies in immunocompromised mouse models. So we have we think we have the ability to now transplant these corrected airway stem cells into at least immunocompromised mouse models. And then I didn't talk about this too much today. So we have considered the safety of editing. So we have looked at the enrichment of tumor-causing genes in the corrected stem cells, and we don't see the enrichment of any tumor-causing genes. And then we also looked at the off-target activity of the guide RNA associated with this correction strategy, and we don't have any reason to be concerned about off-target activity using this guide RNA. And so moving forward, we are thinking about this as a therapeutic modality for treating CF sinus disease in patients not eligible for modulator therapies, either due to their genotype or due to other contraindications. The way we envision this therapy is that the patients would go to their ENT surgeon, where their sinus would get debrided, and then we would get the debrided tissue and isolate the upper airway basal stem cells from this tissue, gene correct them using CRISPR-Cas9, and then the patients would go back to the ENT surgeon after a few weeks, and then the surgeon would debride the sinus again to make space for the corrected cells, and then they would embed our corrected cells in a fibrinogen matrix and administer it to them in the OR. And so we are preparing to approach the FDA to determine what sort of future studies are required for a phase 1 clinical trial testing this approach in humans to treat CF sinus disease. And I would like to acknowledge that this is also an effort that's supported by CFRI, and so we're very grateful for their support. And with that, I would like to acknowledge my mentor, Dr. Matt Porteus, for his incredible support over the years, and then the rest of the CF team in the Porteus lab. And this, as I mentioned, is an incredibly collaborative effort spanning multiple labs at Stanford and across the US and the world. And these are all our funding sources. And of course, we're all thinking about how we could use this strategy to treat CF lung disease in the future. So I will continue to work on that, but I will be moving to Nationwide Children's in Columbus, Ohio, and in a few days, and I will be hiring scientists of all career stages. So if you're interested, please feel free to reach out to me. And with that, I am open for questions.
Thank you. Thank you. That was a whirlwind presentation of your work. It was very, very cool.
Thanks.
Do we have any questions to be fed to me from the chat? And I don't see any right now. So let's see, what questions did I have? How are you going to carry this forward working from Ohio? That's my main question.
Okay. Yeah, no, so the CF sinus trial is going to be led from Stanford. So Dr. Porteus and Dr. Naik and the rest of the CF team they're going to be mainly taking over the CF sinus disease trial. I'll continue to be involved in it, but my efforts would be focused more on taking this technology and applying it to the lung.
Mm-hmm. Yeah, well, that was my other thought was, so correcting sinus disease is fantastic. Of course, everyone wants their lungs corrected.
Yes.
So how do you envision getting or do you envision a similar process of taking cells out, correcting them, putting them back in? And if so, how would you manage the putting back in part?
Yes. So obviously, I mean, I completely recognize it's sort of a tall order, but I think one approach would be to sort of stage it, right? Like, so you start with the nose, and so maybe you try to move down to the trachea or the mainstem bronchi and see what sort of correction or what level of restoration is sufficient in either of these regions in the airway, and whether that's sufficient to improve the phenotype that you see in the lungs. Right. And so you can imagine that if you correct like a portion of the bronchial epithelium, perhaps, then you have enough chloride secretion that loosens up the mucus a little bit, that it improves the outcomes in these patients. Right. So one way of sort of testing this is to just inch forward little by little to see if we can replace at least the large airways first, and then assess how we could go into the small airways. So that's kind of one way we're thinking about that.
Okay, cool. Okay, now I do have a few. Andrea Carter wants to know, will they do this in adults? Oh, the sinus studies. Is this planned, I would imagine, in adults, not children?
Yes, so the phase 1 clinical trial, I think would initially involve mostly adults. I think there's a plan to include a few pediatric cases towards the end if the adult patients are doing well and it's safe and everything, but yeah, it's going to be mostly in adults.
Okay. Okay, Dr. Jonas wants to know, says, amazing talk and exciting work, Sriram. Maybe I missed it, but I'm curious whether you need to control the biodegradability of your scaffolds.
Yes, so that was definitely a factor. So if you look at our cell stem cell paper from 2020, we had started with a porcine small intestinal mucosal membrane, and we don't talk about it anymore because we tried it in the in vivo studies and we weren't seeing that great of an engraftment. And we think it's because the membrane was too thick. So there's definitely a sweet spot. The nice thing about fibrinogen was even in vitro, at the concentrations we were using, the gel was gone within 24 to 48 hours. And fibrinogen has like pro-angiogenic properties and pro-regenerative properties. And so those were sort of the additional factors we considered when we when we decided to go with fibrinogen, right? And it's also degraded endogenously by your body, right? You have an existing pathway to degrade fibrinogen. And so those were all the factors that made us go with fibrinogen.
Okay. Let's see. Krista Icing says, I've had over 17 sinus surgeries. When might this be trial get? She wants to know sort of a timetable. Everyone always wants to know when can we do that?
So we're hoping, it's hard to give a timetable, but like a lot of it is going to be determined by how the conversations with the FDA go and how much further studies they want, right? So we are hoping it may be in the next few years, but I hope, you might have to stay tuned for maybe next year's edition of the CFRI research conference to have a further update on when that trial might be coming up.
Yeah. Can you talk a little bit about how this pre-IND thing with the FDA goes? Just relating it to what has happened in other disease entities?
Yes, I definitely can. Although I don't have personal experience in going through one, but there have been other sort of similar trials that have gone through from Stanford. So one was for sickle cell disease. And so here they showed proof of efficacy in vitro and then proof of safety in vivo in mouse models. And then the combination of those two were sufficient for the FDA to then agree to a phase 1 clinical trial. Obviously, there were other sort of studies done to ensure that they could manufacture a sufficient quantity of cells, and then they could do this reproducibly in a manner where the product is generated in like a clinically safe and GMP compatible environment and all of that, right? So that is an example of a therapy for sickle cell disease. And I think I think the Stanford one had extra data from an in vivo model, but the one from CRISPR Therapeutics, I believe they had in vitro proof of efficacy and in vivo proof of safety. And so our hope is that the FDA would agree to a sort of similar level of proof. If the conversation goes into needing like testing this in an animal model, like the CF pig model or the ferret model, then that might prolong the timeline for the phase 1 trial.
It does. If the FDA does say that we need a large animal study first, how long do you imagine that prolonging the timetable?
It might be a few years. I want to say like at least 2 to 3 years, because it's going to require generation of completely new reagents that would be compatible with the pig or the ferret CFTR gene. And then, so that's, you have to go back to the drawing board to make new reagents.
Right. Okay, Jessica Scarthy asks, how will the corrected cells be embedded back into the sinuses in the patients?
Yeah, so the idea is, again, they would go into the ER with their ENT, and then the ENT surgeon is going to debride their sinus epithelium, and that debrided tissue is going to serve as our source for stem cells, and we're going to correct that. And then the ENT surgeon is also going to receive the corrected cells, and they're also going to get the fibrinogen matrix. And then the patient is going to go back to the ENT surgeon. And then so they're going to have a second surgery to get their sinus epithelium debrided. And then after the second surgery, the surgeon is going to insert the corrected cells along with the fibrinogen gel into the sinus. So the fibrinogen is going to hold the corrected cells in their sinus for a couple of days and give them time to migrate out into the sinus and reform that epithelium.
Okay, and you probably said this, but I missed it. The time interval between the initial debridement and then the going back in and reimplantation.
It's probably going to be a few weeks, would be my assessment
Okay.
right now.
Okay, let's see. Do you need to, sorry about my dogs. Do you need to clean out the bacteria first?
Yes, that is definitely a factor that we're considering very seriously. So there's probably going to be, like cleaning, but then also like some course of antibiotics and things like that to make sure that there's no sort of bacteria in the bloodstream and things like that.
I muted my dogs and myself. Isn't it true that within these sinus surgeries, they debride the sinuses and then they inject antibiotic?
Yeah, I think I'm not a clinician. I think that's standard practice, but we might have like an extra course, or we might add or something like that. So that's going to be defined a little bit more. I think we need to talk to Dr. Naik a little bit more to get the actual details of how that part is going to be structured, but there's definitely a lot of thought going into preventing any sort of infection in the area when we're transplanting the cells, because that's also not a good thing when we're transplanting. Like, it's not a good thing for the corrected cells, right? You want to give them a clean environment to colonize so that they have the maximal chance for successfully engrafting in that tissue.
Mm-hmm. I don't have any more, but now I'm just kind of thinking of them off this spur of the moment. So let's say, you're correcting the sinus cells, and then you move down and try the trachea. Do you kind of do serial biopsies or something to figure out like how far distally the correction has moved, and then kind of inch your way down the airstream that way? Or like, how would this work?
Yeah, that is a good question. That is something we have to think about a little bit more carefully, right? So I think in the sinus, the plan would be to do like a brush biopsy or something like that, and you can probe for the fraction of genomes that have the CD19 tag or the corrected cDNA tag, right? And so you could just use like regular genomics methods to estimate the fraction of corrected cells. So you could potentially do like a brushing of the trachea at different levels, or the bronchi at different levels, which is all routinely done anyways. So you could sort of evaluate something like this in those patients, right? I would say this, that like the tracheal, like going into the trachea or the bronchi is, several years down the line, right? Like those are probably going to be done in like animal models and things first, because you're going into a riskier organ.
Let me check just to see if anyone else type in your questions. We still have a few minutes if you have anything else for Sriram. Okay, if not, thank you very much. I just want to first thank you, Dr. Vaidyanathan, for your very good presentation and your exciting news, and congratulations on your move, and good luck. So that will conclude this session. Please return back to the auditorium at 5:15 Pacific or 8:15 Eastern for our award celebration, where we're honoring 4 heroes from our community, including our volunteer, professional, and inspirational adult with CF, while our friend Paul Quinton will present the 2022 Research Legacy Award. So grab a glass of wine or whatever you want. and join us for a very special event. And then after the awards celebration, the fun will continue when Emily Schaller and Roxy F Foundation will host a virtual dance party. So please join all of us. Well, not me, my back won't dance, but I'll watch. And you can dance like nobody's watching except me. Before the award celebration, we encourage you to visit the booths in the exhibitor hall and take some selfies in the photo booth and post them on social media or chat with friends in the lounge. Remember, the additional chat feature that Siri spoke earlier about has been added. So if you want to talk with friends, you can do it there. And the first research presentation tomorrow will start at 9:15 a.m. Pacific, 12:15 Eastern. So come back and hope to see you then. Thank you very much.